Okay, it is 12 o'clock Central, and we will go ahead and get started. We very much appreciate everyone for joining us for our very first webinar in this series that we're going to conduct at the National Center for Cannabis Research and Education here at the University of Mississippi. My name is Robert Welch. I'm the director of the center here, and we are very pleased and delighted to welcome two prominent speakers that have published a very important manuscript. They are with the FDA, and we're looking forward to their presentation today. Let me go ahead and introduce the topic. I think you probably all know the topic, but just to reiterate, cannabidiol and liver enzyme level elevations in healthy adults, a randomized clinical trial, which was published in JAMA Internal Medicine this year, very recently, actually. And so let me go ahead and introduce you to our speakers today. So we have Dr. Pablo Salcedo, and Pablo obtained his BS in biology and chemistry from Montclair State University and his medical degree from Rutgers. He trained in internal medicine. Previous research areas include pulmonary rehabilitation at the Veterans Health Administration and hypotensive shock at NIH. Pablo also serves as Division of Applied Regulatory Sciences Clinical Trials Team Lead and is involved in the design, execution, and analysis of DARS-led clinical studies. And so then we also have Dr. Jeffry Florian, who's going to join us at the end, along with Dr. Cassandra Taylor as well. So Dr. Florian serves as the Associate Director in the Division of Applied Regulatory Sciences, DARS, Office of Clinical Pharmacology, Office of Translational Sciences at CDER, which is the Center for Drug Evaluation and Research, as we all know, and has worked at the FDA for 15 years. And Dr. Cassandra Taylor has worked at the FDA for over 10 years, and she is the public health advisor at the FDA within the Office of the Center Director. She leads cannabis scientific and research portfolio for CDER and serves as a cannabis subject matter expert within CDER and across the FDA. So we have a brain trust here today of folks who are very knowledgeable about cannabidiol on this topic. And so we're looking forward to this presentation. And I'm going to kick it over to you now, Dr. Salcedo. I appreciate it. And if you need anything from me, just text me or send me a chat there. Thank you so much. And we will have time at the end, folks, who are on as attendees for a Q&A. So you can just please type your Q&A questions there in the Q&A field. Thank you. It's all yours. Thank you so much, Pablo.
Thank you for that introduction. Okay, so I'll be going over the results of an FDA recently completed clinical trial entitled Cannabidiol on liver enzyme level elevations in healthy adults. Disclaimer, this presentation reflects the views of the presenter myself and should not be construed to represent those of the FDA. So for agenda, I'll start with background and objectives. I'll go over the study design and rationale. I'll go over some of the details on the enzyme elevation criteria and evaluation, the results themselves, and then a recap of the study summary and future impact. So starting with background, for the cannabis sativa plant, we have hemp versus marijuana. For hemp, this is a cannabis plant with less than 0.3% THC content on a dry weight basis. This is the primary source of the cannabinoid cannabidiol, or CBD, which is the cannabinoid of interest in our study today. So this cannabinoid doesn't produce an intoxicating effect. It was legal federally since the 2018 Farm Bill, and it's used in a variety of consumer products. Marijuana, on the other hand, is a cannabis plant defined as greater than 0.3% THC content on a dry weight basis. This contains the psychoactive cannabinoid THC, gives you that characteristic high effect. Marijuana remains federally illegal. It's classified as a Schedule I substance, and its legal status varies by state for medical and recreational use. So looking at the CBD market and its growth over the years, so CBD sales in the US grew from a little over 100 million in 2014 to over 500 million in 2018. Current projections put the market at $2 billion currently. According to a 2022 National Survey on Drug Use and Health, about a fifth or 20% of adults reported using CBD products within the past year. In another survey by the National Opinion Research Center, they also saw similar results with about a fifth of adults reporting past year CBD use. So when it comes to CBD safety in 2023, the FDA issued a statement regarding the use of CBD, raising various safety concerns. And this is especially with long-term or chronic use. It noted that studies have shown the potential for harm to the liver, interactions with certain medications, and possible harm to the male reproductive system. So Epidiolex is the only CBD drug product approved by the FDA, and it's indicated for the treatment of pediatric seizures associated with one of three conditions: Lennox-Gastaut syndrome, Dravet syndrome, tuberous sclerosis complex. Those are all genetic conditions. From the Epidiolex prescription drug program, their safety findings, they found transaminase elevations greater than 3 times the upper limit of normal, and 3% of participants given 20 milligrams per kilogram per day. And then in all adult cohort study, 13% experienced elevations greater than 3 times the upper limit of normal. So far, recently completed FDA study, it was a randomized, double-blinded, placebo-controlled study, approximately 200 healthy participants. Data sources that helped inform our design included the aforementioned Epidiolex prescription drug program, an open-label cohort study, and as well as surveillance data. So for our objective, it was to characterize the effects of daily cannabidiol use at consumer relevant doses on both liver enzyme elevations and endocrine measures. So looking at our study design and rationale, at the top left, you'll see self-user reported data from Brightfield market data. So this is over 5,300 consumers. And you can see from here that 84% self-reported consuming 21 to 500 milligrams of CBD per day. And then on the upper end, 12% reported consuming 201 to 500 milligrams daily. This data is also consistent with other identified self-reported usage surveys. Regarding previous clinical trials that helped inform our study duration and sample size, we had 2 key studies in healthy adults that showed liver enzyme elevations. And the first, healthy adults were given 300 milligrams per day for a duration of 4 weeks. They saw liver enzyme elevations greater than 3 times the upper limit of normal in 6.8% of participants. In another study with healthy adults, they were given a much higher dose of 1,500 milligrams per day, same duration of 4 weeks, and they saw liver enzyme elevations greater than 5 times the upper limit of normal in 31% of participants. And it's worth noting that in both of these studies, the elevations occurred during 2 to 4 weeks of duration of exposure to CBD. So looking at our study design, we had 2 arms. We had our CBD arm. Participants were given about 350 milligrams of CBD per day. That comes out to 2.5 milligrams per kilogram twice daily. And that arm consisted of 150 participants. And then we had our placebo arm with 50 participants. Our primary endpoint was the percentage of participants with an ALT based off consensus criteria, or AST, liver enzyme elevation greater than 3 times the upper limit of normal. And for consensus criteria, it's defined as 33 units per liter for males and 25 units per liter for females. So whenever we say the upper limit of normal, those are the thresholds we're referring to. Our secondary endpoints, our secondary liver endpoint, was the percentage of participants meeting withdrawal criteria for potential DILI, or drug-induced liver injury. And then our secondary endocrine endpoints, the first was the change from baseline in total testosterone and inhibin B in male participants after CBD administration compared to placebo. And then our other endocrine secondary endpoint was the change from baseline in TSH, thyroid stimulating hormone, total T3, and free T4 after CBD administration compared to placebo. This schematic is more of a temporal overview of the study design. And so participants received cannabidiol or CBD twice daily for a duration of 28 days. And weekly liver lab assessments were performed. And so these included the entire gamut of ALT, AST, GGT, lactate dehydrogenase, ALP, total DILI, and INR. You notice though, participants had inpatient clinic visits every 7 days, so starting in person day 7, day 14, day 21, as well as day 28. And then we had a follow-up visit on day 35. CBD dosing occurred on days 1 through 28. And then we also collected endocrine and reproductive labs on day 1 and day 29 of the study. And so assessment regarding potential cases of DILI or drug-induced liver injury. These were the 4 criteria that were used to determine if a participant met potential DILI. And so either one of these 4 would meet the requirement. The first was an ALT or AST greater than 3 times the upper limit of normal, with any of the accompanying following: fatigue, nausea, vomiting, any abdominal tenderness or pain, fever, rash, and or eosinophilia. The second criteria was an ALT elevation greater than 5 times the upper limit of normal in isolation. The third, an ALP or alkaline phosphatase elevation greater than 2 times the limit of normal with elevations in GGT. And then the final criteria to meet potential DILI was an ALT elevation greater than 3 times the upper limit of normal with accompanying bilirubin concentration greater than 2 times. And so in our study, participants met either of the first 2 criteria. So what was done as part of the workup when we did encounter a case of potential DILI? So the participant was immediately discontinued from CBD dosing, and then they were closely monitored. And so monitoring occurred every 24 to 48 hours. Repeat liver enzyme collections. These occurred every 1 to 2 days following a discontinuation until they were downward trending, and then they were followed up weekly until the liver function test resolved or stabilized. As part of the workup, participants had an updated history taken to identify any potential exposures, drug use, or relevant contacts. A physical exam was performed and CBC was obtained as well. And then lastly, a comprehensive viral workup was performed in each instance, and that included all the viruses listed below to make sure we accounted for any confounding factors in the viral sense. Okay, so turning more to the study overall and results. So our study began on February 8th, 2024, completed July 17th, 2024. So we actually enrolled 201 participants, 112 males, 89 females, 188 participants completed the study. We had 13 early discontinuations due to non-safety events, and we had a total of 8 liver enzyme elevations. And so as mentioned, we had 8 cases meeting our primary endpoint of an ALT greater than 3 times the upper limit of normal, or an incident rate of 5.6%. And then for our secondary endpoint of potential DILI, we had 7 cases meet that endpoint, or an incidence of 4.9%. And this was, both of these occurred in the cannabidiol arm. We had no elevations in the placebo arms. If you look at the table on the right, so we have listed out and from highest to lowest are 8 elevated liver enzyme cases. So you'll see they range from a high of 18.2 times the upper limit of normal down to 3.1 times the upper limit of normal. You'll also note that the 5 highest cases all occurred in females. And so for these schematics, we're looking at the temporal relationship of CBD dosing throughout the clinical trial. So to orient you, the y-axis is just the times the upper limit of normal. The x-axis is the clinical trial day. The green shading are dosing of CBD. And then our green line is ALT. Our gold or yellow line is AST. And so you'll notice in all 4 of these cases, these are our 4 highest cases, the liver enzyme elevations occurred near the third to fourth week, corresponding with days 21 to 28, towards the end of CBD dosing. And they did follow the same trajectory, so they tended to peak the liver function enzymes within 1 to 2 days and then resolve within 1 to 2 weeks after discontinuation of CBD. And then another incidental finding was we did observe eosinophilia in 7 out of 8 of our elevated ALT cases. In this snapshot, we're just showing kind of the clinical picture that was collected for each elevated case, zooming in. So this is our highest elevated case of 18.2 times the upper limit of normal. And so you'll see the other liver function tests on this graph, including alkaline phosphatase, DILI, as well as eosinophils and neutrophils. And so although not as drastic or significant as the ALT elevations, they also followed a similar trajectory of peaking within 1 to 2 days after discontinuation of the CBD, and then resolving within 1 to 2 weeks. Pertinent medical history for this participant, she did have a history of fatty liver. This was marked non-clinically significant prior to the study. She did report visiting a relative at a farm, so she had farm animal contact. She had no other confounding factors. This was our only participant to report a positive physical exam finding of epigastric discomfort upon palpation. And her viral workup was largely unremarkable, just positive for a past non-active CMV infection. And so this is a type of data we collected for each of our 8 elevated cases. Okay, so recap. So our randomized control trial had a duration of 4 weeks. We administered CBD 2.5 milligrams per kilogram daily, twice daily. We looked at liver and endocrine hormones, liver function tests and endocrine hormones. And we studied a dose on the upper range of what self-reported users of CBD noted. We enrolled 201 participants, 188 completed the entire study, and we had a total of 8 cases of liver enzyme elevations. And then, again, all of these occurred in our CBD treatment arms. And so primary endpoint had an event rate of 5.6%, and then potential DILI event rate of 4.9%. It's worth noting that these results are somewhat in line with a previous open label study that had similar event rates of 6.8% elevations in participants greater than 3 times the upper limit of normal, and 3.4% for elevations meeting potential DILI. We didn't talk too much about the endocrine hormones. That's largely because we didn't observe a significant change from baseline in any of them. So that included total testosterone, inhibin B, and the various thyroid hormones. We call those, that was our secondary endpoint. So just a little bit of discussion on study impact and some insights. So as noted before, our greatest elevations, they occurred in females. This does reinforce reports of increased female susceptibility to transaminase elevations in the literature. It's also worth noting that participants largely did not observe these changes in liver function tests on their own because they're lab values. And so this highlights the need for caution, potentially routine monitoring in CBD users. Also, regarding the lack of effect on endocrine hormones, this might be attributed to the limited duration of monitoring, which is only for a month, 28 days, or the relatively low CBD exposure compared to the reference toxicology studies. Also, the event rates that we observed in our study would most likely be a conservative estimate, given that we only used healthy participants without any comorbidities or concomitant medication use. And we do know that it's not representative of the general population, so that could underestimate risk. But this study was successful. It largely met its goals, and it will help inform discussions about safeguards and oversight of CBD products with the ultimate goal of protecting public health and safety. I believe that is the end of the presentation portion.
Oh, that's great. Thank you so much. Very informative and helpful. And Jeff and Cassie has joined us now, and I've introduced you. I think I mentioned that, Dr. Taylor. If you guys want to come off of mute or on camera, that'd be appreciated. And now we will do our Q&A. If anyone has typed in any questions, I'm not seeing any at the moment. But please just here we go. Okay, so we have one. So this attendee would like to know. I'm just going to read it. You chose to administer 350 milligrams per day, even though your background data suggested that most people use 100 milligrams per day or less. Are there plans to do additional studies with lower doses?
I can go ahead and say some things. Pablo, you can do some of the routing of questions as they come up from chat.
Okay.
So I think the short answer to that is, like, at least from our end, we weren't planning to do additional studies, but certainly additional research is warranted. I mean, the doses that were picked for this study were intentionally selected to be towards the upper quartile of what was reported use, and really to go and help move the conversation, which was that they're not necessarily like any concern. Like, you have one side saying nothing really to worry about at doses that are being used, and another side saying, well, here's the clinical data at these much higher doses showing what was seen. And that represented a gap in the information. So this was overall intent, trying to pick a relevant dose and move that needle a little bit closer. Additional research would certainly be warranted at lower doses, looking at populations who may be older, maybe taking additional medications, something more representative of real world use, so to speak. There'd also be value in looking at longer duration of usage. And so all of those are probably additional investigations that would and hopefully will be done by some groups.
Okay. I
Yeah.
see.
Oh, go ahead. No, Pablo, if you
Oh, no.
wanted to add anything, I just had one little quick thing to add there too, is the fact that, um, as everyone knows, I mean, when you start trying to ascertain what kind of dosing you're getting in a real world kind of setting, real world evidence type study, I think it would be important to look at some of those quote real world products that folks are out there potentially using as well, which complicates everything because most of the time we don't know what exactly is in a lot of these products that are being sold and consumed, either by consumers or patients, whatever you want to call those folks that are taking these products. And so it kind of bleeds a little bit into this next question that was asked by someone, is where did the CBD come from? If anyone wants to.
Yeah, Pablo, do you want to answer that or?
Yeah, so the CBD was a pure product, prescription drug product that was obtained from the manufacturer. So it was 100% CBD.
Yeah. And
And it was oil.
just adding on to what Paul was saying there, it was a prescription drug product and was intentionally picked that way. We understand well aware that some of the consumer products may have various contaminants that could also contribute to a safety signal. The intent of taking the prescription drug product, take something that was known to be pure, wasn't going to have impurities, so that whatever would be seen from the clinical study would have a cleaner interpretation attributed to the drug itself as opposed to one of the potential contaminants.
Yeah.
Yeah. And we use the drug product also because we were dosing, healthy adults, and a lot of that data hadn't yet been in the scientific community. So we understand a lot of products in the marketplace are not necessarily the same purity level, but this is really to help create a foundation for additional studies as Jeff had previously mentioned.
Excellent. Yes. And I think this would be a quick one. So the route of administration was oral, correct? Yes. So that's an easy one there. I'll mark that one done. And then there was one question about measurement of free T versus total T. Do you have any insights on that?
Yeah, so we measured TSH, total T3 and free T4. And so for the total T3 being combined and uncombined together, this was largely in line with previous studies in the literature. And so we followed suit on what was typically collected to compare the endocrine endpoints. But there was not a particular reason to choose total T3 over free T3.
And I think this one question I'm seeing now, I think you just kind of alluded to that about the DILI cases with CBD are marked from oral CBD oil. And so we just answered that. So that was the case with this study, is it did use the CBD oil. I think that helps to clarify that. And then there was a question about the impact of CBD on female hormones, estradiol and FSH. And if you have any insights or thoughts about maybe exploring that further or looking into that more, or is that something you guys would like for folks to maybe explore?
Yeah, I can start with this one. So the main reason why for the reproductive hormones we focused on male hormones was because we based off of reference toxicology studies on which we largely saw changes in male from animal studies such as decreased spermatogenesis or decreased testicular size. And so that's largely the motivation for looking specifically at the male reproductive hormones.
Very good. Yeah. And so, with the possible 5%, with the possible DILI cases, that 5% with the elevated enzymes, were there any age or gender correlations, or is there any plan to look at predisposing factors like genetic polymorphisms in liver enzymes?
I can start with that one as well. So at least demographically speaking, there wasn't any pattern. The participants were the entire age gamut of a healthy adult, 18 to 55. I did see another comment on other relevant medical history. The example
Yeah.
we used in the study, we used that one because that was the most significant positive finding wise. But all the other participants had negative confounding factors when it came to their medical history, physical exam, contacts. For the viral workup, there were maybe one or two more that had non-active past viral infections like Epstein-Barr virus, but those were all resolved or non-active at the time of the study. And so the most significant medical history would be of the one we shared.
Yeah, and I can add a few items. So we did see the imbalance with more events and higher elevations in women compared to men. The study wasn't powered to look at what would be sex differences, but there's definitely an imbalance that stood out when we were looking at the events. Warrants looking into further. I think there was another comment about looking at maybe genetic polymorphisms for metabolism. We did collect drug pharmacokinetic data as part of this study. Would mention that it was more trough data at the end of each week. There wasn't really any difference that we saw with regards to those measures. Looking at CBD or the hydroxy and carboxyl metabolites, no difference in the PK levels per se between those with events or without events. All the exposures that we saw for this population were in line with what would be expected with this amount of dosing for this duration. So we didn't really, our take was this wasn't necessarily driven by differences in PK exposure, though it's clearly we know that it is related to the drug exposure itself, and like higher doses having more events. I think this jumps into some of the like next questions there. Based on the timing of the events, the fact that you don't really see elevations occur necessarily, like in this data, or even from let's call it the clinical trial program data within the first week or so. The interpretation from that is what's driving the elevations more likely than not to be one of the metabolites. A lot of times the carboxyl gets implicated from various valuations, but just that timing of onset is suggestive that it may not be CBD exposure itself, but maybe something that takes some time to accumulate in tissues. And we do know that the carboxyl has much higher exposures with repeated dosing. I'll say one other item. We are looking, one of the teams within our division is doing an omics style analysis, trying to understand, I think it's with proteomics, looking at was there any notable differences among those participants who had liver enzymes elevations or eosinophilia compared to on CBD compared to those without, and also using the placebo cohort for comparisons. I know those evaluations are ongoing. Given the population size of only having 200 total, and then seven or so individuals with events, what a type of interpretation one might be able to make from any of the findings that are likely to be exploratory, it probably highlights another area where there could warrant additional investigations to maybe understand what exactly may be predisposing factors um, um, for increased risk. Because again, like not everyone is having elevations, but if you can flag those who might be at more risk, that would be an important thing to do.
Yeah, and I think that kind of addresses the next questions at the top there that I haven't flagged yet. Do the other participants with elevated ALT, AST have any medical history, just like the one with the fatty liver and cytomegalovirus infection? I think you kind of just addressed that. And then the next question, I think we've already hit on this, is talking about it asks which CBD did you use and how does it, now this is the kicker for all this stuff is the second part is how does it compare with what is used typically in, let's say, 1,000 milligrams at night. I'm assuming that is referring to preparations that are found over the counter, so to speak, or at convenience stores, online, gas stations, gummies, and solutions and so forth. And that's the issue that we face with all this, is that a lot of those products are available out there and there's no real good way to compare something somebody says they're taking that they bought, and they may buy something totally different the next month when they run out. It's really difficult to ascertain exactly what's in those products. This is why it's so important that what was used in this study was used, because it's purified as much as you can possibly get. So we have the answer, because that other stuff, there may be something else in there, a byproduct or an impurity that's causing issues versus the actual CBD itself. And some products that we've tested, we did a study, I think a couple of the scientists are on here that ran this study. We published a paper about some of these products that were obtained online, and some of the products didn't have any CBD at all in them, and the label says, "Hey, this is X milligrams of CBD." But I think you get the point to that. Let's move on to the next. Okay, I think we've already kind of hit on the fact that I think you've addressed the fact that the liver enzyme elevations didn't emerge for 3 to 4 weeks. And then the second part of that is, would FDA allow clinical studies of shorter duration than 3 weeks to move forward without the requirement for monitoring liver function under a clinical IND?
Yeah, I can take that one. So, That is a review question. Someone would have to come in and they would have to present their study design. This is not the forum to be discussing specific study designs and what's appropriate. So someone would have to come to us with the IND and it would have to be reviewed, and that would have to be a discussion, I think, for the review division. Because of course we have to make sure that the patients are going to be safe in whatever study design, dose, duration. There's a lot of factors that go into those safety decisions for an IND. So
Yeah.
we can't give any answer there at this time. Please, you can look at the guidance that we have about INDs, or if you don't have it, we can share it with Dr. Welch and he can share it with everybody as well.
Thank you, Cassie. I appreciate that. And the next question addresses looking at the topic of the increased liver elevation in females, and do you think the hormonal status, such as contraceptive or PCOS, could be a factor, and we need to have it as a variance? I think you've addressed this somewhat already. I think we've already kind of covered that. And then the next one is talking about was CBD dosing compliance assessed in any way? So that was just a pretty straightforward question. Did you guys have any assessment of whether patients took their doses appropriately?
Yeah, so we did have, I didn't mention it in the presentation, in addition to checking in on the study site and the in-clinic days, we did have a dosing diary that the participants completed to just note when they took the CBD and just to hold them accountable. And in addition, we did have pharmacokinetic samples taken so that we would know concentration-wise that CBD was being consumed.
Very good. Thank you. And then the next question, I think we've addressed this somewhat. Do you think a PK study would have helped to explain some of the reasons for females being more prone to DILI? I think we've kind of, Pablo, do you have anything to add there?
So I was looking at the next question. So for this one, we did have the PK draws for all participants to look at the measures and the concentrations of the CBD. Regarding this kind of leads into this next question about explaining the difference between males and female elevations. We would, from our study alone, we can't comment on that. But if we were to kind of entertain some speculation, one possibility could be the difference in fat distribution for females. And so we know CBD is fat soluble, could end up being stored to a different degree, but that would just be, a speculation.
Mm-hmm. Very good. Thank you. And then we have the following. For efficacy studies, it is hoped to obtain 80% positive results to allow for parametric statistical analysis. In a safety study, what percentage becomes a significant concern? And I'm assuming they're meaning what percentage increase in elevation.
Yeah, I can go ahead and say some things on this. And so there's nothing really that's going to be said. It depends on the nature of the safety event. I mean, there's certainly a whole laundry list of drugs that have been removed for the market for what are fairly rare but serious events. I mean, something that actually truly, like where you're having liver failure, where you're having torsades, things like that. Don't really so much need powered comparisons between arms, just like occurrence and event rates, like 100,000, 1 in a million can be enough to be something that can translate to labeling or other items. So not really a threshold, and would also say, and this might be an element of what's there. The aim of this study wasn't really to put together any definitive statistical comparisons. One of I know when we were designing this, there were a lot of questions that came up. Would anything be seen at all? If went with these doses, 150 individual, like, could it be 0 events? Pablo went through some very nice items about was literature or some of the clinical study data that was really used to help make a projection, and ended up lining up what we saw in the study lined up very well with that. But there were a camp, probably like internally, probably even external, that doses of this, would anything be seen at all?
Yeah.
Um, it was nice that, well, shouldn't say nice that, but it is nice to have results that help move the needle in terms of what might be seen with doses of this nature. And then really, if one wanted to go and do statistical analysis, really trying to understand differential risk, that's something where we're hoping results like this can be used by other groups to construct such a study and maybe answer those questions.
Excellent, Jeff. Thank you. That's very good. The next two questions are somewhat related, so I'll kind of try to combine these. So, asking about would inhaled CBD potentially reduce these concerns as it bypasses first pass hepatic metabolism, and is FDA aware of any cases of liver toxicity in individuals using CBD via inhaled, or, now this adds, or topical routes, or have all examples been with oral dosing?
Well, I think Jeff and Pablo's study is only for oral, so
Yeah.
their study, it can't correlate to any other dosage forms outside of that. So that's what we're talking about today. And I think I'm not a pharmacologist, so I would think things definitely could be different based on the route of administration. How that impacts metabolism, I think, can become a bit more complicated if you're taking an oral versus a, inhaled route. So
Yeah.
I think there would need to be probably some additional studies done to have some greater understanding of what that might look like, and what that would entail, I think is outside of the scope of just from today's presentation. So we would have to figure out, if folks are actually looking into that.
Mhm.
We're aware that the inhalational route is very common. That's not lost on us. But the study really focused on the oral route, as again, we were trying to create a foundation to gain understanding of the potential toxicity.
And to add to what Cassandra said, there's also a paucity or very limited data and research out there on the oral route. So that also was a motivation. There's quite a few studies that look at the inhaled route of administration, but when it comes to oral administration of CBD, it's more limited.
Yeah, that's a good point. Really, outside of those Epidiolex pivotal trials, there's not a ton of other data out there. So this is very helpful that you guys did this. One quick question about food effect. Was the CBD taken kind of agnostically in terms of before or after meals?
So participants were instructed to take the CBD after breakfast and after dinner. And then when they were in the clinic, they were given the CBD about 30 minutes after breakfast and dinner.
Very good. Thank you. Yeah, that's very helpful to know. So, this question, in addition to the classical metabolites, hydroxy and carboxylic acid, have you considered detoxification of CBD quinone? And that may not be something.
Oh, we don't have our bioanalytical experts here.
Yeah,
Can certainly.
we don't.
Yeah, I'd say like really the focus we had from our end was what was termed here like the classical ones, the hydroxycarboxyl. To whoever asked the question, if they wanted to send any literature Dr. Welch's way to pass to us, I mean, I think we'd be interested in learning what might be the concern or what there might be to learn about it.
He's my colleague, so
Yeah, it's Amar. He can email us. Amar, please email us. It's a good question. We just don't have our bioanalytical expert on the call today.
Yeah.
But I think it's a good comment.
Yeah, it is, Amar. Thank you. Now there is a final comment here. It's not it poses a question. It says Delta 9 or Delta 8. And I think that in terms of this conversation, neither. But a lot of these products that are out there being consumed that are under the guise of being hemp and so forth, and which I talk about ad nauseum, and I uh, am still talking about it because it's still an important issue out there. Is that, you know, there may, in those particular products, as we mentioned earlier, there may be some delta 8, some delta to 9, some other cannabinoids in there that are intoxicating. And so, with this particular study, this once again to reiterate the importance of obtaining the prescription form of CBD, is that it has trace only amounts of delta 9 THC, and that was something that was done through the clinical trial program that the FDA approved, and every batch and every bottle of that particular product had to be specified and standardized. And so these other products are not that way. And so that's what makes studying this kind of complicated in terms of looking at a trial like you guys did such a wonderful job at. You have to obtain something that you know what's in the product. And that kind of bleeds over into, one of the reasons we want to have this series of calls is to kind of highlight issues of clinical importance. I know there's a lot of cannabinoid or cannabis related calls that are out there that go on frequently, that deal with a lot of other aspects of this complicated world that we deal with. But we're really focused on that clinical aspect to try to help providers and also ultimately the patients. I mean, that's what we're trying to really get to the bottom of, is trying to help those folks. And another one final question just popped up that says, are there any plans to look at some of the other minor cannabinoids such as CBG, CBN, CBDV, CBE, etc.?
I think we would love for you to look into those, because I think, we do our best to create the foundational research, but the resources are limited. I don't think anyone on this call doesn't understand that, especially where we're at right now. But that is a very important point. As Dr. Welch has said, there's a lot of products that are out there that may say, CBD in giant 72 font, and then down below in like 9-point font, there's an alphabet soup of other minor cannabinoids, and people are being exposed to those potentially. Yeah. I think we should also say that we're well aware that there's not a lot of truth in a lot of these labels in the market, unless you're getting it maybe from a state-run dispensary where they have a lot more controls versus something that is in kind of the weirder gray hemp space that is largely uncontrolled. And so I think it would be important for folks in this audience, as well as researchers who maybe weren't able to attend, to be able to create studies where they can start to look at these minor cannabinoids individually and in combination, because people are being exposed to them in various quantities. And I know we've said it before, but I'm going to say it again. We don't know what's in a lot of these products. I'm not sure the manufacturers always know what's in a lot of the products. Just because someone doesn't analytically test for something doesn't mean that it's not there. So if you're only testing for 5 cannabinoids, doesn't mean that there isn't 20 more present. And so it's a great question to whoever asked that. I think it's, we look to the research community to try to create studies that could help to tease out, what exposures look like and toxicity levels, etc.
Very good. And then we have one, it looks like a final question has popped in that says, what is the half-life of CBD? I'll mark that one. And a lot of that depends on the route of administration. I know that, it really depends on what you're talking about, because it can range from hours to days. You got to remember that, and I don't mean to answer this question if you guys have anything to add here, but we do know about all these cannabinoid molecules and compounds. They're very lipophilic, and so they they have very vast volumes of distribution, especially taken in the oral route, very, very low bioavailability. And so it can take some days to really get it to steady state. But I didn't know, you guys, if you have a set answer for that you'd like to provide, but please do so.
I don't. I just wanted to add, it's also, frequency of use, strength of the dose.
Yeah.
Those are all factors as well as your route of administration. So I think there's not an easy, single answer
No.
to that question, unfortunately, but I think that just goes to show why, we need more folks to enter the space and to create more research, because it's a challenging space to be in. And especially if you're adding CBD and maybe 2 or 3 other cannabinoids, that's going to impact how it affects the human body and how long it stays around. Are you eating a fatty meal? Do you have low body fat? I mean, there's so many variables here that could impact that. So I don't know if Pablo or Jeff have anything they want to add, but I think the answer is it depends.
Yeah.
Very unsatisfying answer. I'm sorry to the person who asked.
It's true, though. It depends.
Yeah, we're sorry, but do the
I'll
best we can.
So I'll just have one addition to that. Like all of that, this is tied to sort of a second part of the work we did, looking at drug interactions. And so again, like maybe something worth mentioning is cannabidiol and its metabolites also can affect the metabolism of a lot of other compounds in a lot of different ways. It may not always be fully appreciated how that's happening. I mean, I think the Epidiolex prescription drug label really reflects what type of effects it may be having on other drugs. But at least like conversations we have, consumers who are taking cannabidiol not as a prescription drug may not be aware of its potential to have some effects on other medications. I think that's probably another thing important to be aware of. It's why one of the things highlighted that there might be increased risk in a broader population using other concomitant medications, both what might be liver injury as well as what might be effects, increased or even decreased exposure on the other medication. I guess supposed to be increased because it's an inhibitor on those medications. So part by saying that it's one of those, like the information in the prescription drug label is good, but how many consumers are probably looking at that if they're also just regular consumers of non-prescription cannabidiol products?
Yeah, it's a good point, Jeff. I don't think any of those label are passed over to some of these unapproved products. And I think generally the public is rather unaware of those. We try to get the information out there, but it's sometimes challenging when you're up against large marketing firms and things of that nature, and those products are very different.
Yeah, it's a good plug for some of the folks that are helping us accomplish some of what we're trying to do with Dr. Khan's team at the Natural Product Center here. They do a lot of work in conjunction with us to elucidate some of those multiple drug interactions. We're doing some studies now that looking at commonly used over-the-counter products like acetaminophen or ibuprofen, other analgesics that are commonly used over the counter, and as well as other antidepressants that could be used concomitantly. That have some data out there, but it's not as robust as we would like for it to be, because I know that's a big area that patients are using these products to maybe help alleviate some of those symptoms. And so they look to these products for certain things, it seems like, outside of the prescription world, to alleviate some symptoms that they haven't found an answer to in that prescription world. So that's a very good point, Jeff. I don't see any more questions, and we are doing good on time. We got 6 minutes. We have 6 minutes left, so I'll turn it over to you guys if you have any final comments. I will just say real quick that this has been recorded, and so we'll get this to you guys if you're interested in re-listening to it or watching it again. And we will post it to our website as well, and can send you guys a link. But go ahead, please go ahead, and thank you.
Yeah, and thank you. I first want to thank Pablo and Jeff for agreeing to present their research. I think it's really important that they have the opportunity to say in their own words how the study was conducted and the results and the assessment. So huge thanks to them. Also huge thanks to Robert for providing the venue. We really appreciate it. I think everyone on the call knows that sometimes you write a paper and it gets out there and there's always questions. So I appreciate that we had the opportunity to do some live Q&A and answer some of those questions that maybe folks had burning since they've read the paper. And also just a plug that we need more research in this space for CBD, but also other cannabinoids. And so if you are someone who's in the research space or thinking about getting into the research space, if you're not sure who to talk to or who to reach out to, I know Old Miss now has some resources available to researchers, but you can also reach out to us if you need help. We can direct you to people. There's lots of guidance documents. We have had some staff reductions, so sometimes it's a little slower than it used to be, but we really do encourage you to continue to do research in this space. It's very much needed. We cannot do it ourselves. It's a team effort. And so please continue. For those of you who've been in the research space, we thank you and we appreciate the work that you're doing. And we hope you continue to do it to the best of your abilities. And I hope we'll be able to attend the webinar and listen to some great talks in the future, as I'm sure there's lots of other great research out there that we can learn from as well. Jeff or Pablo, anything you want to add?
Nothing else from me. Thank you everyone for taking the time to listen to our presentation.
You did a great job, Pablo. Thank you.
I'd say likewise. Thank you for this opportunity. If there are any questions, please feel free to reach out to us regarding study items. Happy to clarify those post this webinar. Echoing what Cass says, there's a lot more work to be done. We hope some of that research may be done or may already be ongoing from those of you in the audience, and we look forward to seeing about this.
Well, thank you so much, everyone, and we appreciate you for attending as well. Attendees, thank you, and I will be in touch soon for our next webinar. Dr. Vandry has agreed to present next month, and so we don't have an exact date just yet. But I think he was on the call, but just dropped off. So we have all your contact information now, so we'll get that over to you, and it'll also be posted to all of our social media and everything, so you'll know what the topic is and what time and date and everything. So with that, thank you guys so much. Again, we appreciate your collaboration on this and being our kickoff webinar in this series. And have a great week. Thank you so much. We appreciate it. Bye.